This recorded session is dedicated for the post-laboratory for Module Number 2, Soil and Air as Environments. During our last synchronous meeting, we discussed how we will carry out the virtual laboratory activity in order to achieve the following learning outcomes and objectives. At the end of the activity, you, the students, the performers, are expected to first, associate microbial adaptations to the nature of habitats. Second, determine the nature and distribution of microorganisms in soil. Third, you must be able to explain how physical and chemical factors affect the nature, distribution, and diversity of microorganisms in soil. Fourth, you must be able to quantify soil microorganisms using the standard plate count. And lastly, you must be able to isolate, quantify, and compare viable airborne microorganisms using the gravity plate or the settle plate and impingement techniques for the case of um, microbial sampling from air. So if you will notice, the first three are actually outcomes, while the last two are more of the objectives. No? And I hope that as you perform or as you watch the simulation videos, the demonstration videos, and you submitted your worksheet already, you were able to really achieve, especially the objectives. No? The last two, which are the topic learning outcomes on the quantification and isolation part. Like somehow, I hope that you were able to, uh, um, you know, um, um, from the demonstrations, you were able to pick up the key concepts. No? Before we discuss the complete details for the virtual laboratory, we will walk through a set of curated concepts in order to reinforce some key discussion narratives that I want all of you to be introduced to. So let's begin with the idea or the concept of soil genesis and the factors affecting this very important process. You will notice here a formula or a mathematical expression that I adopted from Hans Jenny. He was an expert in soil science, so he is actually a pedologist. No? So, as a pedologist, he formulated a general mathematical expression that tells us the relationship between these independent variables and the formation of soil. The soil itself as a material, its properties, and the process of its formulation or the process of its formation. No? So these some of the independent factors affecting the process of soil formation include climate, the parent material, the topography of the environment, um, living organisms that are found in the environment, in the specific environment, be it macro in micro, and of course time, one of the most important factors. So of special interest to us in the study of microbial ecology are of course the living organisms, both micro and macro. No? Another factor in soil genesis that might not really be present in this, in this representation, in this mathematical expression, is anthropogenic activities such as cropping and cultivation. So, of course, we use soil as a very important um, matrix for us to produce food and to grow or cultivate livestock. So these anthropogenic activities that we practice with the use of soil also affect the properties of soil itself and the process of its formation. No? Okay. Now, let's take a look at the different types of soil, because this concept is something that I want you to understand as early as now. No? One common type of soil are mineral soils, which are derived from weathered rocks and minerals. Mineral soils, they are formed and shaped by the different geological processes of the planet, and they extend from the terrestrial surface up to the underlying parent material, which is usually unaltered or unweathered. No? Later, I will show you a picture of the soil profile of the profile of some certain types of soil, so you can appreciate the appearance on a longitudinal visual. You will appreciate how the soil is actually structured. No, so from the terrestrial component or the terrestrial side, the topsoil, up to the parent material. The parent material of mineral soils, they can be composed of residual weathered material from solid rock. They may also be composed of unconsolidated material, normally coming from one location, be transported to another location via the processes of sedimentation, erosion, or the formation of ice caps for the process known as glaciation. No? So the factors affecting the process the weathering process itself may be physical, biological or biochemical, and chemical in nature, and all of them contribute to the processes, no, to all the weathering process, which will include disintegration of rock and conversion of primary minerals, no, such as feldspars and micas, to secondary ones such as clay components, no, clay elements. In the process of soil formation, the process of parent material weathering, rock weathering, no, we expect that soluble constituent elements in inorganic forms are provided in the soil matrix, or dispersed in the soil matrix, allowing the very one of the very important functions of soil, which is to support life, to support both macro and micro life present in it, no, or using it as an important matrix. Another type of soil which we will talk about, since in our virtual session we included this particular type, though not really this one that I'm going to discuss further, but in relation to water, no? In relation to water, another type of soil is those that form in coastal areas and tidal marshes, or inland water bodies, like the ones that are near surrounding a river or surrounding a lake, or the ones that are in the coastal area. If you've been or if you have used uh, the coastal road connecting Cavite to Manila, probably you've noticed on the side of Manila Bay, the side that faces Manila Bay, you will see a portion of the soil that is rich in plant growth, especially. I'm not sure if it's really rich, but I've seen mangroves, and there are specific areas along that location, that actually have mangrove growths. No? So those type of soil that grow on such watery areas. No? So these types of soil most of the time they're waterlogged with plant residues that are submerged in water for a prolonged period of time, especially the mangrove area. So through time, the accumulation of organic matter results to the formation of what we call organic soils, such as peat, muck, and bogs. No? Like for the case of for the case of inland, no, but for the case of mangrove areas, they have that specific. I can say that it's still organic soil, though it's kind of hybrid, because the water that logs that soil is consisting of salts as well. So it's not purely fresh water. But for the case of inland water bodies, the soil surrounding them normally will become organic type, that are very rich in matter, in organic matter, because through time, there's accumulation of organic material. No? Okay, let's review soil profiles for you to further appreciate what we're talking about. Let's take a look at the next slide. So, the horizons within a soil profile of different types of soil, they vary in terms of thickness depending on the intensity of all the independent variables involved in the soil formation process. No? Though the boundaries of these horizons may not be very clear cut all the time, no, so like what you can see here, it's kind of hard to tell whether there's really an outstanding visual difference in terms of the color or the appearance itself. No, normally the uppermost layers of for the case of mineral soils, they are the ones that are most altered during the soil formation process, while the deeper layers, on the other hand, will hold the original parent material that are normally unaltered or unchanged. No, for the case of organic matter soil, for the case of this one, very dark colored soil. I think this one is also organic matter. You can see the rich, the and this one too. So you can see the growth of plants on the top soil part. So so the organic for the organic soils, no, the organic matter enriched horizons, the upper layers are called A horizons. No, so we call them A horizons, very rich in organic matter. The underlying enriched layers in mineral soils, on the other hand, we call or we refer to them as B horizons. No, so B horizons are for uh, what do you call this, for mineral soils. They are like the counterpart of A. Though for the case of organic matter soil, you may appreciate the presence of or for the case of organic matter soil, we also have that topmost area called the A horizon. The C horizons, on the other hand, are the least weathered part. So this area is the one that is nearing the weathered material. So you expect it to be found on the deepest layer, no? Very deep layer, or the one that has the bedrock material, nearing the what do you call that, the water table, nearing the water table part of the soil. No? And then for organic soils also, we have to notice that commonly they are saturated with water, and they're composed of the plant material we expect to see from them most of the time are mosses and water loving, no, vegetation. No? And um, uh, another important layer that we have to talk about is the upper material called the O layer for the case of organic soil. So the O layer here. So you can also see it's not really for the case of the other soil types found or represented in this visual. It's not really present, but for the case of this one, let's talk about the O layer. So the O layer is actually composed of it's actually composed of what do you call this, uh, um, organic matter that leach from the uppermost portions, from the upper layers, from the A horizons. No? So we expect the organic matter to traverse or to be mostly present on that O layer side. No? Then we also have the vadose layer, which is already underlying everything, all horizons. So this layer is unsaturated. The parent material is expected to be found here downward. Then eventually it reaches the water table, and the soil becomes saturated with water. So you expect, you know, the soil water component to be present in the vadose layer as a material which is already concentrated with water, not very rich in organic matter because it's already very deep, no? And you don't expect the biomass produced by living organisms on the top layer. You don't expect them to leach through the vadose layer. No? So the inherent distribution and containment of nutrients in the different horizons, different layers of the soil are all related to the ecosystem services that we get from them. What is this ecosystem services? Ecosystem services are the different benefits that we humans are provided with by the natural environment consisting of water bodies, and for the case of our topic, for the case of soils, provided to us by by soil ecosystems that are healthy. No? So such ecosystem services that we relate to soil include agro ecosystems, definitely, forest ecosystems, grassland ecosystems, and for the case of aquatic ecosystems, there are some portions that are containing soil as well, like for the case of mangrove ecosystems, hybrid aquatic and soil. No, so the interconnection of soils, nutrient cycling, the very important process of nutrient cycling, biodiversity, and anthropogenic activities, as well as, of course, for our learning for our course, the activity of soil microbial communities. All of these factors, their interconnection is evident in all the benefits that we get from healthy soils, such as for the case of plant production, diba? We use agrosoils that are rich with nitrogen-fixing bacteria to support our food supply and to ensure food security. Diba? We also have soils that are rich with microbes participating in the carbon cycle. So carbon sequestration is an important ecosystem service we get from soil systems. So, then, for the case of biological activity. So soils that house varied life forms, both macro and micro, they provide themselves as a matrix for interactions where gene pools are diversified, and metabolic capacities of the different species are diversified as well. In the grander scheme of things, no, the metagenome of all living systems within the soil matrix are diversified because of the soil, of the health that the health status that soil actually possesses. No, in addition, something that I would like to mention is the different types of ecosystem services as we have defined them, no, a standard definition. One standard definition provided was from the United Nations. No? So according to their Millennium Ecosystem Assessment, they divided the ecosystem services we get from soil into four types. So there are ecosystem services that provide, there are those that regulate, there are those that are for cultural purposes, and there are those that are for support. No? So the ones that I presented here, all of them are, or these they cannot be typified into one particular type because the definition provided by the Millennium Ecosystem Assessment is more of very ano siya, very human-centered, very anthropogenic. No? And for our case, of course, we're not really adopting an anthropogenic lens whenever we talk about soil microbes and soil microbial communities. No? Okay, so indeed, we can really say that or we can remember that when soils are healthy, a lot of ecosystem services are delivered to us. A lot of ecosystem services provided by the soil enable the life in our planet. So everything from food production to climate control to carbon sequestration, all of them are dependent, and all of them are ecosystem services provided by soil. No? So, what we are highlighting in our module are soil microbes. Diba? So, soil microbes, as a highlight for for our discussion, I think I've mentioned some of this, or I've already talked about them during our last synchronous meeting. We talked about the different categories of soil bacteria, especially soil bacteria. Kasi for our case, focus of most of our activities will be on bacteria, fungi that are found, no, in the different matrices. So let's recall those bacterial groups that we divided into four categories. So decomposers, most of the bacteria found in the soil are decomposers, and they consume simple carbon compounds, no. uh, for example, the exudates coming from roots and plant litter. Those are examples of organic matter that decomposers can consume. So in this process, bacteria convert energy in soil organic matter into forms that are useful to the rest of the organisms within the soil food web. No? So a number of decomposers may also be able to break down recalcitrant material or recalcitrant components of the soil, such as fertilizers, though not all the time. Though. So, in the process of decomposition, there is immobilization or retaining of nutrients in the soil, primarily because the cells make use of them. The bacterial cells make use of them, and then the other living the other living components of the soil will also make use of these um, nutrients that are processed or broken down by soil microbes, by soil bacteria to be specific. No? So the processes of nutrient cycling, storage, decomposition, as well as water movement and disease suppression, all of them are related to the activities of decomposers, no? Of bacteria that are categorized as decomposers. A second group of bacteria are the mutualists. No? So they form associations or partnerships normally with plants. And the most well known of these mutualists are the nitrogen fixing bacteria, which I have been discussing in the lecture and in our laboratory sessions. No? One good example of nitrogen fixing, or there are a lot, no, but later I'll show some or I think I've shown in the lecture naman a visualization, no, of how soil bacteria associate with plants. So we know that for the case of nitrogen fixing bacteria, since nitrogen is a element that's very or molecular dinitrogen is very hard to break down. We need the I don't know, the activity or the bacterial activity of these nitrogen fixers. No? A third group of bacteria in the soil are the pathogens. No? Bacterial pathogens such as Pseudomonas, Agrobacterium. They can cause changes in the morphology of plants, no? For the case of pathogens. And then we also have lithotrophs or and chemoauthotrophs. Okay, so this group obtain their energy from compounds such as nitrogen, sulfur, iron, or hydrogen instead of the carbon compounds. No? Instead of the organic compounds, they get it from those inorganic ones. No? Some of these species that are categorized as lithotrophic or chemoautotrophic, some of them are also important in nutrient cycling, especially nitrogen cycling. No? So if you notice, all these four groups, no, they contribute to several ecosystem services. These bacterial functional divisions, they all contribute to different services that the soil provide. No? So here I've shown this picture last time. So some more examples in terms of, for us to visualize. No? So we have actinomycetes here, and then we also have Azotobacter, which is a nitrogen fixing bacteria. It can associate with plants, and then later on it can also, or it functions or it does the most important function of nitrogen fixation. No? Okay. All right. So for our module, for the activities that we visualize, that we watched using our demonstration video, so they are divided into three. So first, we did soil pH measurement. Second, we did soil moisture or water content measurement. And lastly, we followed a demonstration video on isolating and quantifying soil microbes. No? We worked on especially bacteria and fungi. These three things that we did. Let's before we take a look at all of the results, expected result, no, let's discuss a few concepts on each of these components. No? Let's take a look at each of them. Let's begin with soil pH. No? So for soil pH, the natural or the parent material that is responsible for the production of the soil actually plays a very important role in dictating soil pH. No? Of course, in supplementation by weathering processes, as well as other factors, climate, your vegetation, topography, and time. So all those things are important for the determination of soil pH. But generally, soil pH acidifies through time. So you expect the pH of a soil to acidify through time. Why? Acidic protons that are supplied to the soil from the atmosphere, from organic sources. These protons react with bases such as the aluminosilicates, the carbonates, and other mineralogic materials, as well as the humic components of the soil. So those acidic protons, they interact with those basic components of the soil, and given sufficient time and water, precipitation, basic cations will be exchanged from mineral and organic constituents by hydrogen. And that will reach throughout the surface of the soil, increasing or decreasing its pH, acidifying it. The presence of clay materials or clay minerals, which are saturated with basic cations, they retard the process of soil pH lowering. Um, but we expect that through time, as the factors continue to play, then you add anthropogenic activities like agricultural activities, you will definitely accelerate, no, or ensure the process that soil will be acidified in no time. For the case of semi-arid or arid, very dry, no, regions, they lead to an opposite trend. The soil will be buffered at an alkaline or basic pH because of the presence of sodium. So sodium is an important element for soil that are found in semi-arid or arid areas. So the pH for that type of soil we expect to be more basic, no, more alkaline compared to the usual soils that we work on. In terms of nutrient availability, pH influences the quantities of nutrients that are soluble in soil water. Some nutrients are more soluble if the pH is acidic, while others are preferring more alkaline settings. No? In general, soil pH affects soil microbes by affecting the activity of the enzymes, the soil enzymes. No? Remember that enzymes are proteins that are very sensitive to factors such as temperature, pH, and any sudden change on these factors will cause conformational changes to soil enzymes, affecting the activity of soil microbes as well as other living organisms in the soil. No? Um, we can measure soil pH. Traditionally, what we do to measure soil pH is to prepare a soil paste, normally via the addition of a diluent, such as a calcium chloride solution, and then we use an appropriate electrode. No? But before we proceed, with measuring pH, I think something that I did not really mention is how we get soil, how um, how we process soil. So there are two types, no, of or there are there. Oh yeah, there are two types of soil sampling. No, you can directly get, or you the first one, which is not really scientific, is you just get a sample in your particular land or particular soil area. You get one sample and then you prepare it to you make a soil paste from it, adding a diluent. Though that's not very systematic and it might result to bias readings, some practice that because of ease and because of the fact that it will take not so much time. The ones that we expect to do, like if we did a face to face meeting, is composite sampling. So composite sampling will require accumulation or sampling of soil from the different randomized representative plots within your land. So you get, let's say, if you have a land that is too big, you can apply different sampling methods based on your need, based on your objective. So a randomized composite sampling will require the combination of several soil samples from the different locations of the plot, and after combining them, you do the pH measurement. So that's the more scientific way. Like if we did this in the lab, that's what we are going to do. No, composite sampling. Now going back to pH measurement. Um, while the process is relatively easy, the process of reading the pH, we must remember that interpretation is quite difficult, especially if we want to associate it with microbial processes. Why is that? Because the concentrations of the cations that are sorbed or present on that are kept in the top surface, on the top soil or the surfaces of the um, soil sample that we are going to process. No? So the concentration is actually 10 to 100 times higher than those of the soil solution. So that there is bias, or there is already an inherent difference. So that's something that you have to put in mind. So all the more that you really need to use composite sampling instead of just the traditional direct sampling, where you get some soil from a specific plot, and then you do your soil pH measurement. No. Okay, so the interpretation part when it comes to pH, in pH measurement, pH reading is going to be quite tricky, and we need to factor other characteristics or other expected phenomena happening. No. Okay. One of the types of soils that we worked on, or that we intended to work on, is acidic, no, acidic soil. So acidic soils, they have pH values that are less than 6.5, because normally the expected neutral pH of soils should be between 6.5 to 7.5. Anything below 6.5 can be considered acidic, and those that are lower than 5 are very acidic. No? The process of acidification, as I've mentioned already, is dependent on the breakdown or decomposition of organic matter, contributing to contributing acidic protons to the process, or to the soil matrix. Other factors aside from decomposition of organic matter that affect the lowering of soil pH include chemical fertilizers, such as ammonium-based fertilizers. They are actually major contributors to soil acidification. Ammonium nitrogen is readily converted to nitrate and hydrogen ions once they are already present in the soil. If nitrate is not taken up by the plants, then that's a problem. It can leach away from the root zone of the plants to the soil component. Then the hydrogen ions also leach, and they can increase acidity. They can decrease the um, they can decrease or lower down the pH value, the pH measurement for the soil. Also, we have agricultural use, so plant production and animal raising, okay, affect acidity of soil. So most plant material are alkaline in nature, and if they are removed by animal grazing, or ones that you have done your harvesting, no, of course, what you leave behind are the acidic components. Also, another factor affecting what they call this another factor affecting the lowering of pH is the introduction of calcium and magnesium, normally during precipitation, during rains and floods. No, in terms of um, in terms of uh, preference, no, for growth, some plants actually prefer acidic soils, which we term as sour soils, over alkaline soils, which are termed as sweet soils. Normally, ornamental flowering plants, they prefer highly acidic soils compared to the neutral ones, or compared to the basic ones. The overall acidity of the soil will actually affect nutrient availability. No, so all major plant nutrients, nitrogen, phosphorus, potassium, sulfur, um, and also the trace elements such as molybdenum, they may be unavailable. No, in very acidic soils, these major nutrients may not be available, and they may be available, but in very low quantities, very insufficient quantities. Plants that grow in acidic soils will definitely show, no, symptoms of the effects of acidity. Let's take a look at how soil pH will influence healthy plant growth. Let's take a look at this picture here. So figure 1 shows us the relationship between aluminum concentration and the pH of the soil. So you expect that acidic soils will harbor or will allow aluminum concentrations to accumulate at up to more than 5 ppm, which is already very toxic to even tolerant, no, aluminum tolerant species. And then as the pH increases, we expect aluminum not to be concentrated to such amount, so lower than 2 ppm, and it's already considered non-toxic. So the more acidic the pH is, the more aluminum concentration there is in the soil. And we expect that will definitely affect the growth of plants, like what you can see here. So here is barley grown in acidic subsurface. So the one that you can see on this side are those that are grown in basic or not very acidic soil. Aluminum concentration that is lower than 2 milligram per kilogram. So you can see the roots have grown longer than the ones that are here, the ones that are grown in a pH of 4.0. So the roots have grown smaller or shorter in terms of size. Okay. Aside from acidic soils, we also worked on waterlogged soil. So this is another type of soil that we dealt with in this activity. Waterlogging occurs when roots cannot respire due to the excess of water in the soil profile. And normally, this is caused by a drainage issue. So waterlogging is a drainage issue. The water does not necessarily have to be present on the surface for you to say that waterlogging or the soil type that you're working on is a waterlogged type of soil. Okay, so the major problem with waterlogged soils is oxygen. Very low concentrations of oxygen in waterlogged soils. Also, waterlogged soils will increase will release increased amounts of nitrous oxide, which is a greenhouse gas that we don't want to be produced, diba? Or we want to control the concentration of. Due to excess water, oxygen diffusion in waterlogged soils very slow compared to those that are not waterlogged. So that meaning your waterlogged soils are poor in terms of free oxygen, very poor in terms of free oxygen. As in itself, no, water is really a very important component of the soil. It's worth an examination on its own. That's why we dedicated a separate section for that. So for soil water content, we know that soil water affects the available moisture to all the organisms living in the soil matrix, and as such, no, we have to factor in what things affect soil water content. Okay. We also what enumerated these components as the following. So the aeration status, nature and amount of soluble materials, osmotic pressure, and soil pH. All of these things contribute to a retainment of water, of moisture in the soil. No? So, um, okay. So in relation to microbial ecology, to the study of microbial communities found in the soil, of special significance to us is the fact that water in soil is absorbed strongly to itself and to the surfaces of the soil particles, normally via the action of hydrogen bonding and dipole forces, dipole interactions. No? So that means that water is not going to, water will have a different behavior in soil. No? As it is absorbed in soil particles, it will remain unfrozen even if temperatures reach below 0 degrees Celsius. So it becomes very available. No? Now, as a reminder, no, soil water content may be measured in a mass or volume basis. So we can measure soil water content via mass or via the volume component, no, or via the volume unit. Uh, I'd like to stress more about the importance of soil water, no? Kasi soil water in as a important component of soil, if I remember, water acts as a transporting matrix. So it transports, it flows, and as it flows, it transports materials from one place to another. So for the case of soil water, no, that function is very evident. That from one location to another, organic matter transport is facilitated. One of the agents that facilitate it is water. Also, as a solvent, diba? Water is known as the universal solvent, and hence it serves as an important reactant in chemical reactions, and as well as biological reactions within the soil. Okay, so because of such importance, now we have to take note of how soil water content will affect the properties of the soil sample that we're working on, as well as the activity of the microbes present in the soil sample. Okay. So here, for the first part of the module, no, you are tasked to compute for the percent moisture content, as well as to speculate on the pH values of soil types according to indicated description. No? So we have acidic, non-acidic, waterlogged, and non-waterlogged soil. So I think the fresh weight samples and the oven dry weight samples of the soil are given, and then here you're asked to um, speculate on the pH of waterlogged as well as non-waterlogged. So for waterlogged, we may expect that the pH is below 7. Any value that's below 7 can be acceptable. For non-waterlogged naman, the neutral value 6.5 to 7.5, or the basic greater than 7 is also acceptable. Then for the moisture content computation, you know the formula. You just get the difference between the fresh weight and the oven dry weight. Then you divide by the oven dry weight. Then you get it in percentage. So you have to multiply by 100. These are the values that we are expected to see as your answers. Okay? Now, a second important component of the virtual laboratory session is the isolation and quantification of soil microbes, particularly bacterial groups and fungi. No? Before we discuss the expected results, let's walk through some key concepts. No? We can classify soil microbes metabolically into phototrophs and chemotrophs. So we have phototrophs and chemotrophs. So remember that phototrophs use light as energy source, while chemotrophs, on the other hand, use chemical forms of energy other than light. No, so not light, not other than, but not light. Doesn't use light. It uses chemical energy, no chemical forms of energy. So these fundamental two groups can be subdivided or can be further categorized into organotrophs, which uses electron sourced for for organotrophs are organic. Then we also have lithotrophs, in which the electron source used is an inorganic source, and for aquatrophs that make use of water as their electron source. No, so here we can see some examples, representative examples of electron sources. Okay, for the case of a photolithotroph, reduced minerals. For photoorganotroph, organic carbon-containing compounds. For photoaquatroph, we have water. And for chemolithotroph, we have reduced minerals or inorganic ions. For chemoorganotroph, we have organic carbon compounds as their electron sources. Then for electron acceptor, you can see here um, some representative elements or acceptors. Then we have for carbon sources, okay, some other examples of carbon sources. Okay. Connecting the microbial groups together, the ones that we've talked about, no, is an electrical system of electron flow, which consists of two cycles, an oxygenic or oxygen-producing cycle and an anoxygenic or a non-oxygen producing cycle. It's composed of four connected circuits that are hooked together in a parallel fashion. So one circuit is considered as chemo organotrophic. No? Then we have phototrophic circuits, dalawa, two phototrophic circuits, and then we also have one chemolithotrophic circuit now. So three groups of organisms are responsible for carbon addition to the soil matrix, and only one, the chemoorganotroph, are the ones responsible for the removal of carbon. So phototrophs, photolithotrophs, chemolithotrophs, and photoaquatrophs, these three groups introduce carbon in the soil matrix, while chemoorganotrophs are the ones that proceed with the removal. So their activities intend for the removal of carbon in the soil matrix. No? These two cycles, we distinguish them on the basis of their photosynthetic mechanisms. For the case of photoaquatrophs, we expect oxygenic processes. No? We expect oxygenic events, oxygen producing. While for an oxygenic cycle, no oxygen is involved. No oxygen is produced, no, in the process. Okay. Now, let's take a look at some of the soil bacteria that we can expect to isolate from soil samples. So if we really did our activity face to face, we can expect to see some representative soil bacteria. These are actually some of the more popular examples of soil bacteria that are expected to be present in the soil. So we have nitrogen-fixing bacteria, those that have symbiotic associations with plants, especially the legumes. When they associate with plants, they form visible nodules And then what they do while they reside in the plant material, they supply or they supply nitrogen. No, they convert nitrogen into a form that will be usable to the plant, while the plant provides them with carbon compounds. Then we also have nitrifying bacteria, which are responsible for changing ammonium to nitrite and then to nitrate, which is a preferred type of nitrogen for grasses and most row crops. Nitrifying bacteria, they may be suppressed in forest soil so that most of the nitrogen will remain as ammonium for the case of forest ecosystems. Then we also have denitrifying bacteria that convert nitrate to nitrogen or nitrous oxide. Denitrifiers, they function without oxygen, and as such they are saturated in soils or in inside soil aggregates, so they can be found in compact soil that should be free of oxygen. So for them to work, for them to be capable of doing their job, they must be secluded in an area of the soil that is lacking oxygen, that will be free from oxygen access. No? Then we also have actinomycetes, which are a large group of bacteria that grow as hyphae-like fungi. They are responsible for the earthy smell of uh, of of the soil, especially when rain comes. No, you might smell geosmin, or your in Tagalog the alimuum scent, that's actually the earthy smell produced by actinomycetes. No? So actinomycetes, they have a variety of functions. They can do decomposition of several substrates, but most importantly, they have been eyed to participate in the degradation of recalcitrant compounds. Now, very hard to degrade materials such as chitin, cellulose, no, normally will require activities of these actinomycetes. No? And some actinomycetes are also capable of antibiotic production for the case of streptomyces. All right, so let's take a look at some pictures, what we are expecting to see if we grow them in soil culture media. So we have rhizobium, okay, nitrogen fixer. Then we also have normally the nitrogen fixers. They look like normal bacterial colonies when you grow them. So they will appear as such, very normal bacterial colonies. Then we also have actinomycetes. No, so here I'm showing you some plates of actinomycetes, actinomycete isolates, probably streptomyces that I have isolated myself a long time ago, probably 7 years ago. No? So actinomycetes, they will look like this when you grow them in yeast agar, no, in yeast extract agar. They will look leathery in appearance. The colonies are showing as powdery, leathery. If you like open the plates and you will whiff a scent of the geosmin. No, you will definitely smell that earthy smell for actinomycetes. If you look at them at the microscope, they will look like this. But they will form strings, no, and they are gram negative, of course. I mentioned a while ago that some of them are capable of antibiosis. No, some of them are capable of producing antibiotic compounds that fight other pathogens, other microbes. No, so here I did a perpendicular assay. So I from one of the isolates I grew on one part, and then did a perpendicular streak of the different microbes, no? I think Staphylococcus, Escherichia, yeast, Candida albicans, no? And you can see there's a zone of clearing here. You notice. So this zone is an area where your actinomycetes secreted secondary metabolites that are anti or against the other uh, microbes that I grew on the perpendicular position or the perpendicular orientation. Okay? All right. So much about soil bacteria. Now let's talk about soil fungi. Soil fungi are one of the focus organisms in our module. They are microscopic. Normally, they look like the cells look like plants. They grow in long thread-like structures known as hypae, and that together, when they grow as big in terms of volume, will become macroscopic. Will become the mycelium. No, though that's for the case of molds. No, while other fungi such as yeast, when you grow them in agar, in plates, they will appear as what do you call this, bacterial like in terms of colony appearance. Okay, so for the case of molds, we expect the macroscopic mycelium to be the uh, nutrient absorbing component, or the nutrient absorbing structure. While for the case of yeast, for the case of yeast like Candida or Saccharomyces, no, they may have very different structures. Now, for the categories of soil fungi, we expect to isolate. One component or one category are the decomposers or the saprophytic soil fungi. Saprophytic fungi, they convert dead organic matter into fungal biomass, as well as carbon dioxide and organic acid. So they secrete exoenzymes that will degrade the dead organic material. They're essential for the decomposition of cellulose material or cellulose-based materials, very woody material. Okay, your soil fungi are responsible for that. And by consuming the nutrients from the dead organic matter, they play a very important role in in nutrient retention and immobilizing the elements in the soil matrix. Second group of soil fungi, similar with soil bacteria, we also have symbionts. So we also have mutualists. A good example of soil fungi that is mutualist are the mycorrhizal fungi. Mycorrhizal fungi are the best known soil mutualists. They grow inside plant roots, like the behavior of nitrogen fixing bacteria, and there are several groups of mycorrhizal fungi. So what they do, they increase the surface area of the root so that they can get more nutrients from the soil matrix. And then the last group of soil fungi that we may be able to isolate if we did this activity face to face are the pathogenic soil fungi, like the ones from the verticillium, the Phytophthora, Rhizoctonia, and Pythium group. So all of them are pathogenic soil fungi. So let's take a look at some pictures. So here we can see mycorrhizal fungi, the white ones. These are the mycorrhizal fungi. They increase they increase surface area of the root for better absorption. Then here is a representative example of a soil fungi affecting living matter or living tissue of a plant, allowing it or leading to its death, eventually to the death of the living material of the plant. Okay. All right. So now let's talk about the activity proper, which is on the quantification, the one that will somehow the component of the activity that is left for you to understand, left for you to perform, is on the quantification part. No? So there we have two formulas for, or we have two formula for computing standard plate counts for soil microbes. If you have two consecutive tenfold dilutions that have valid counts, you use this formula. Okay? Where summation Z is sum of all colonies in all the plates counted. So I ask you to consider 3 plates. No? Like for the case of the ones available in our in our module, no? So the colonies represented there are the average numbers, no? Though I'm not sure if that's how you did it, because probably some of you did a different manner, so or did it differently. So we'll talk about that later. But for the purpose of our post lab, the summation C is sum of all colonies in all plates counted. So if you expect two dilutions, so you'll have 6 plates. So all these 6 plates you have, if they have valid counts, you compute or you sum all the colony counts. No, so you get all the sum of the colony counts. And then FW is the wet weight of the soil divided by 1 times N1. N1 is the number of plates in lower dilution counted, so that's 3 for our case, plus 0.1 times N2, number of plates in the next higher dilution counted. So N2, that will be 3 as well. Then D is the dilution from which the first counts were obtained. So, depending on which is available in our worksheet. VP is the volume plated since we did spread plating, so that should be 0.1 ml. And then ODW is a dry weight of the soil. Okay, so if you have 2 consecutive tenfold dilutions, you can get valid standard plate counts for your soil microbes. If you only have 1 plate or 1 dilution that has counts within the countable range, then you use a different formula. So the only thing different here is instead of using the dilution, you use the reciprocal of the dilution. Okay? Some reminders. Okay, so some reminders in terms of standard plate count. We have varying standard countable range. So some say some protocol will follow 30 to 300. Some will have 25 to 250. For our case, I think I've mentioned that we follow 10 or 10 to 20, and then or 10 to 30, and then uh, um, the upper range, no, the 250 to 300. So it will depend on your purpose. So you don't have to be, specific on what is a countable range, because it will depend on the protocol. It will depend on the sample and what are you trying to count, what are you trying to isolate and count later on. So, but normally, as a rule of thumb, 25 to 250 is mostly used. Then the second mostly used countable range is 30 to 300. Okay, so for the bacterial counts, you are tasked to you are tasked to fill out the blanks here, diba? So we use 10 to the negative 4 and 10 to the negative 5 dilution. Okay, so you expect that, of course, 10 to the negative 5 will have lower counts than 10 to the negative 4. Okay, so it will be dependent per student. And then for the standard plate count, I developed a calculator for that. So once I check your computations, I will just input whatever you followed or whatever you included in the blanks, and I will get the standard plate count that is expected. That's for actinomycetes, the case is also the same. Expected that 10 to the negative 4 will be having lower counts than 10 to the negative 3. And then the standard plate count will be computed na lang automatically. And for fungal counts, the same thing is expected. Okay, 10 to the negative 3 values, the counts will be lower than the ones in 10 to the negative 2. Okay? And the standard plate count will be automatically computed na lang din as follows. Okay? So, in terms of soil type, no, do we expect to see a specific dominating group? Generally, soil will have most actinomycetes. No? We expect actinobacteria to be present in high concentration, in high representation in soil, while bacteria and fungi may have a different distribution, varying distribution, depending on the soil type. But as a rule of thumb, remember that most soil, 10 to 15% of soil, is always dominated by actinobacteria. No? In fact, we call actinomycetes or actinobacteria as soil bacteria most of the time. So if you will read through some more literature, you will notice that actinomycetes are called soil bacteria because they are the ones that are highly dominant in in most types of soils. No? For the case of bacteria and fungi, distribution and dominance is multifactorial. Okay. I will choose to stop at this slide before we proceed to discussion for Module 2 Air. No? So we'll stop with this slide muna so that we can upload this PowerPoint, this recording easily. Okay?